Mark is an associate professor here at BCS, McGovern Institute. So he's a fantastic biophysicist, and he has done really, really cool work on electrophysiology. Previously, he was in general pharm, and now he has done lots of neat work on human cells also. He found very interesting features of how human pyramidal cells, are different from other type of species. And he's going to join us today to giving us his perspective about what he heard today and the challenges that exist from the point of view of an experimentalist and a biophysicist, and how the sort of efforts that are ongoing and we heard about today could help move the field forward for someone like Mark, or from his collaborators. For example, he works closely with Sid Cash, and Shadi Dai actually today talked about the collaboration with him. And we have very complex systems. For example, you measuring activity of with thousands of neurons, whether it's with UT electrodes or you have a dense microgrid on the surface, but deep down underlying we have these biophysical elements that are doing the actual computation, and also they go offline and they go crazy, and then we have seizure. So Mark is going to tell us about all those. And Joe is a computational neuroscientist by trade, and he was previously at Center for Theoretical Neuroscience in Columbia, and then also at Johns Hopkins University. And lately he has joined NIH Brain Initiative as a scientific program manager, and discussing all these topics with John and Guy. So he's a good representative of what we want him to go back to, basically to the office of the Brain Initiative and discuss with them in terms of what is the perspective of the people who are trying to create the tools, and what are the perspective of the people who are generating the data, and what's the perspective of people who are trying to analyze the data. And we will see what they have to say about this. And then after the initial discussion, then we open the panel, we open the questions to the public. So everybody who has questions from the previous speakers, feel free to ask them, and we'll continue from there. We're supposed to end at 5:30 because we'll have a reception, and we don't want to keep people from reception. So please.
Thank you, Nima. I don't know how we should go about doing this, because I'm not sure how interested all of you are in my particular perspective, even though I do seem to have a connection to many of you here. I was at Janelia with Tim and others. Nice to see you, Tim. I was a next-gen leader at the Allen Institute, so I was visiting all of you at the Allen for a couple of years. Matteo, I've actually tried to reproduce one of your experiments, and we should talk about that. It went pretty well. So there's a number of you that I have connections to, whether you know about it or not. As Nima said, I was trained as a biophysicist. I do a lot of ion channels and synapses and synapse physiology, but I do systems neuroscience as well, which is how I wound up doing this one experiment, some of which is in humans, some of which is in rodents, some of which is comparative work across a lot of different species. And reproducibility and what doesn't reproduce across species, across areas, across laboratories is near and dear to my heart. So I appreciate the opportunity to be involved in Odin. So I could talk forever about this, but I don't think that's as interesting as maybe facilitating a more involved discussion with my colleague from the NIH about all the different things we've heard about today, because it has been a very wide-ranging discussion with a lot of pretty serious implications for how we think about the future of neuroscience and how we think about the future in our own laboratory. So I, at lunch, went up and talked to some of the members of my laboratory, some of whom are sitting back there now, in a pretty impassioned way, I think they would say, after the beginning of our symposium here, about how important these issues are to thinking about their futures and the future of what I do in my lab. And probably by extension, many of you have had the same kinds of thoughts. How do we think about the opportunities, the burdens, and the challenges of applying these new technologies that folks have shown us from, experimental design hardware, imaging, optophysiology, electrophysiology, and then all of these great software tools for trying to build pipelines and stuff? How do we manage these obligations? I think that's really what we've been sort of struggling with. How do we think about what users ultimately want? How do we produce tools and data out there for people to become users? And so I think that's probably the kind of stuff that we want to center our discussion around. Would you agree?
Sounds good to me. Can you hear me? Is this working? Okay.
I don't know. I can hear you. I don't know if they can.
Good.
Yeah.
Okay.
So how do you think we should go about this? Because I thought it was great. You know, we clearly all had a lot of questions earlier for all the speakers and in the panels, and I think we kind of want to just reinvigorate that, unless you want to, you know, riff on some of the bits and pieces I brought up or things that other folks mentioned, particularly from the NIH perspective, which I'm sure all of us would love to hear.
Well, okay. For those of you who don't know me, I am, I guess, the data sharing guy at the NIH Brain Initiative. The last guy was thrown into an alligator pit, and so they hired me. This kind of feels like I'm in the middle of well, at the other end of a firing squad. And I sympathize, and I've heard your pain, and I have lots of perspectives. I would like to be asked questions, but I guess at a high level, I'm interested. Well, kind of what you've said, and basically thinking about electrophysiology as opposed to something like the BiCCN or BiCAN projects, where there's kind of there's the CCF, this common coordinate framework that's integrating all this predefined modalities that are being, and there's projects to co-register, and there's all this coordination. If we're thinking about neuroscience broadly and the Brain Initiative more broadly, then we have this diversity of modalities and EFIS, diversity of modalities and behavior. How do we actually, what's the most useful way to quantify behavior? We're standing up this new BBQS program to answer some of those questions, but there's tons of uncertainty about how to do this in a way that actually integrates usefully into knowledge in a way that we want to induce kind of superlinear interactions and generate lots of good science in a very kind of energy efficient way. And so the diversity of modalities is a problem. But then there's also the whole metadata question. So Jim DiCarlo mentioned suggested that the metadata problem might in fact be impossible to solve. And it seems both impossibly critical and important to all of this, to everything that we're doing, but also it may be impossible to solve. And it's unclear. We can all just, everyone can be recording more and more of the characteristics of the data, maybe surreptitiously hiding microphones in the ceilings and whatnot, and leading to who knows what kinds of problems, or what kinds of t-shirts everyone is wearing, and maybe color matters. The impossibility kind of seems to come from the whole endeavor of science is we're always figuring out what are the latent unknown factors in the background context of experiments that are kind of affecting the endpoints and the measurements and the experiments. So as science happens, you learn more context, you learn what's important, and then you've already recorded data, without controlling for those factors that you've learned about later. And so there's always this problem of like the historical trailing, a lack of control for unknown contextual factors, which is a fundamental problem with data. I mean, so there are some contrarians out there who say that data is maybe more of an obstacle generally than it is a boon or a benefit to science. And so that's kind of like an extreme end of...
Wait. Data is an obstacle to science? That's a serious contrarian.
Well, if we think about data, I'm not saying that's me. There's, on the site formerly known as, there have been arguments made, but there's we're thinking about scientific ideas versus scientific data. We want to collect data in order to both generate new ideas and to gain confidence in those ideas, and to also drop ideas which are not good. We want to prune and evolve the set of ideas. We want to better understand the possibility space of science going forward, and data helps us do that. But we need to help data help us, and so that's what all of the infrastructure, all of the talk about governance, all of the bottom-up constraints and top-down constraints as far as the technology and storage problems, and then the metadata problems and everything. And now I'm blabbing.
No, this is great. This is exactly what we want. So how do we help data help us? That kind of could have been the mission statement for the conference. I mean, that's kind of perfect, right? So I think that's exactly where we should be going. Matteo has something he'd like to add.
Like a totally side view. A word that I haven't heard for the whole day is animals, and how sharing data could reduce animal use. Maybe somebody has done an experiment that is not exactly the one you want, but if you have their data, you could get close to where you want to be. And then if you have some refinements you need to do, then you have to use animals. I haven't heard it mentioned even once. And so, and I think it shouldn't be part of the equation, that's all. I don't, we don't want to steer away. You had a really good direction. But
No,
just
I think
added it
I think this is exactly what we want in the discussion. I mean, there's some threads, some overarching threads, right? We can keep pulling on, but I also think we want to think about complementary or potential synergies here, right? I mean, I think certainly as far as the NIH is concerned, and as far as those of us that are worried about animal welfare are concerned, right? That's a big upside of opening a bunch more data and putting a bunch of it out there. Cataloging where these advantages are, I think, is part of our mission here at the conference. I think that's great. Ben.
One perspective I'd just like to bring up, and it connects to this reducing animals and also the what metadata we need to store, is actually kind of starting at the finish line and then working backwards, which to me is actually thinking about someone really reusing a data set that's been shared, right? What do they need, like training people on how to do that and then learning from them, figuring out where their pitfalls are, and then trying to translate that back to what kind of metadata we need to store, what kind of infrastructure we need to build to facilitate the data access, to find the necessary data sets, what kind of training materials we need to create in order to allow people to get started in that process. And we're starting to build this, with our team, we're starting to, we have this event now we've run for 2 years with Neurodata Rehack. We have a small group, we're trying to facilitate this. The Open Scope has the data book, which is helping training materials. But I think what is kind of free form right now, and maybe you could use some processes around it, is really figuring out where the pitfalls are when people do try to do the reuse data, and somehow translating that back to guiding the infrastructure and guiding the submission of data all the way down to like what metadata we need to store.
Yeah, I think policies and processes to help do this. Like we don't know what we don't know till we try it a couple times, and then have someone who's basically naive try to go in and test a hypothesis in that data and see where all those potential breakpoints, failure modes, stoppages, et cetera, occur. You have to choose, what experiment are you going to use to do that in? Is it something that's attractive enough that folks will really passionately want to reanalyze it? But I think building test cases like that are really tremendously valuable. I thought it was one of the things that was really great to hear about the OpenScope project and the IBL today is prior to those presentations, we were thinking about like, okay, more data, produce more data. How are we going to deal with 2 terabytes an hour, thousands of labs, thousands of students and postdocs? Right? It was a little overwhelming. In the way we think about free-form investigation for a whole bunch of different laboratories. But in terms of these really focused examples about how folks went about prosecuting specific structured investigations, even if in the case of the IBL there wasn't a specific hypothesis to test, it was still highly structured. Tremendous amount of value there, I think, for us thinking about how to do this in a way that is going to be more open, inclusive, effective for reanalysis. Maybe how to comply with FAIR, like et cetera, et cetera. I thought that was really great.
That's a very useful spectrum to think about things. Sometimes I think about things in experimental design in terms of like empirical content. Like basically how many bits of entropy do you need in order to define like what the task is here? Like how many joints are there at which you can carve the data? And so that's totally up to experimental design. It's up to degrees of freedom. It's up to the animals that you're working with. That also depends highly on the scientific question and kind of the maturity of the field within which the scientific question is posed. And so if data and modeling and all this is really here to increase knowledge and accelerate the rate of knowledge production from data, then the context includes the scientific context, which is a problem-solving context. What does everybody know? What's the consensus in the field? And how does that inform the interpretation of data? So if a great data set's collected and it makes a seminal contribution to a field, and then it gets a secondary use 10 years later, that scientific context, in a sense, is a social context that doesn't exist anymore. And so there's almost no way to perfectly recreate that, right, unless somebody really digs into, like, okay, let's read all the literature from 10 years ago and to understand the context in which this was created. But then there's like resources where maybe there's lower empirical content, but what you're trying to do is really map out comprehensively, some space of behavior or brain activity in a way that like the IBL is doing or some of the larger brain projects are doing, and that can be more kind of closer to the objective end and the less objective end, where there's less context dependence about data. Sir, you have your hand raised.
There was a lot about data reuse, right? How do we make data reuse more convenient and useful? In neuroimaging, human neuroimaging, surprisingly, we came to two types of experiments which are most reusable. Resting state, when it's like you do nothing, right? And actually rich stimuli, where people watch movies, which are really nicely controlled and everybody goes through the same emotion. So we have two spectrum, kind of end of spectrum, and in the middle we have nicely controlled experiments. And those, actually, on the end of spectrum, they are mostly more reusable than anything else, right? Because those are tightly controlled for specific design. Other such types could be discerned in animal research. We have just freely behaving, animals that could be something like resting state, from which people could discern different, aspects, right? If they have sufficient description of what led to those behaviors and sufficient capture. Other rich stimuli, which could be then collected and lead to multiple reuses if we have sufficient metadata.
Audience, what do you think? Mackenzie.
Yeah, I mean, kind of piggybacking off of that and some other comments that were said, I mean, one thing that sort of struck me is we talked a lot about data, but we didn't talk about why we're collecting it. Like, I think the biggest problem in the field is the lack of theory, not the lack of data right now. So to me, I'm a little bit worried if we just collect more data, we continue to stamp collect, but we're not actually testing things that are agreed upon in the field. So in that sense, I agree with like the open scope and the way that it's being done at the IBL. I'm sure they had a hypothesis. I imagine at some level, but.
Can I ask a question to your question though? I mean,
Yes.
you saw a graph of how many people have downloaded the ALED data. Presumably they were testing something. I don't know what they were writing about. They must have been testing something, some idea, some hypothesis.
Oh, hypotheses, yes, but I'm saying like real theories, like theory-guided experimentation is I think something that we're lacking in the field. Like coming to the fact of like petabytes of data at CERN that they're collecting to, yes, but they're also, they know what they're looking for. Like the Higgs boson, they knew what to look for in the data, so it was clear what the aim of that experiment was. I don't think we have that in neuroscience very cleanly yet.
And I totally agree. I mentioned the Higgs boson earlier today to someone else in a very similar conversation. So I'm not just the data sharing person. I have a theory background and a computational modeling background, and I completely agree because the theoretical framework within which you're working totally informs the context about why you design an experiment the way you did or how you interpret it, right? And so it's clearly that's what we want to be doing here, is extracting knowledge within a theoretical context in order to understand.
I wanted to continue on that idea, but first to answer your question, so the main importance of the ALED data, right, at least in my experience, though it might be in many other people's experience as well, it's less to do experiments on it, but more to use it as a sort of way to annotate other experiments, right, like to constrain regions of interest in analysis, right, because it's still data, but it's a tool sort of data more than a subject sort of data. But on the topic of data reuse, so somebody asked, I think it was also you, right? Like if we look at reducing the numbers of animals used because somebody, instead of doing a new experiment, will find a different experiment, there's a huge problem with that, which is twofold. First of all, the data record might be too hypothesis heavy. People tend to describe what they wanted to do more so than what they actually did, which is a problem. The other problem is if you look at effects, we live in a highly aspirational world, I think, where we believe that, the distribution of sample sizes across levels of different variables doesn't matter. But if you look at a question for which the samples are unbalanced, you can still do your statistics, but they'll be adequately formed. Which is why I wanted to bring up perhaps the easiest way to do reuse is to not start with the naked data, but simply with a study, with an article, with a finished output, which is completely re-executable. What do you think about that notion? Because in that case, all of those interesting reviewer questions, for instance, what would have happened if you varied this parameter? What would have happened if you did this other thing? They would be automatically addressable because the entire article could reproduce itself directly from the data. And then incrementally, you could start addressing the new question simply by deriving what you already know works. So sort of battery included approach. It's also more accessible. right? You're not just you and the data and, oh my God, what do I do with it? But you have a pipeline which works, which you can then just derive.
I would just say this excludes review of questions which require you to go and collect new data.
Yes, of course.
So that's a core tenet, right, of reproducibility, where basically that's the ideal case. We want to be able to get to the point where maybe every paper that's published, I think even eLife and some other journals have like, live, I'm not sure they call it live papers, but
They have live segments.
live segments where there's executable code essentially, and basically, maybe one level upstream of data processing to allow you to compute those kind of like final statistics or figures or whatnot, and for comparison. And that kind of gets at, what are we doing reuse for? There's a reuse for reproducibility, which is to say, you're checking that last mile of the analysis that everything checks out. There's also replication, which is where, okay, we do actually want more of that kind of like end-to-end pipeline from like raw bits all the way through to those final figures, in order to get kind of more confidence in the results that have come out of those studies and out of that data, but then also to allow basically branching. So once you're replicating, then you can also branch into new science, and that's critical for discovery, and so we need both sides of that, where you're basically reproducing from one level up in terms of processing, but then also going n levels back towards raw data, and then as far as NIH perspective, brain initiative perspective, we want to enable both of those. But I mean, that really depends on what's the goal, the specific aims of the grant that gave rise to those papers. What's the goal of that particular paper? I mean, so what I say is it's up to PI discretion to a certain level to find that sweet spot of that trade-off between what is the scientific accomplishment or scientific advance that's being reported. What was the goal there, and then how can you best enable that to contribute to future science?
I think we had an urgent question out there, or comment.
So I was waiting for someone to start this discussion. We've had many discussions that artificial intelligence is waiting to be unleashed once the data is properly structured, and both for data queries, but also for suggesting analysis and even closing the loop to steer the experiments. And so I know a lot of work is already being done on language-based interactions with the data, and so how will that change the landscape of reusing of the data if we can interact with it with richer tools, enabled by AI. So we have many discussions here, but I think I'd like to hear more.
Yeah, that's a great direction to take things. I mean, it's getting better at writing code. I think most of my students and postdocs aren't writing their own code anymore. I think they're asking ChatGPT to do it.
Suggesting visualizations for
Yep.
different types of data. It's just like, okay, have these types of series. What's the best way to visualize it with our limited dimensions?
I think it's a great direction to take things.
What's
We
the-
haven't really talked about it thus far.
I was just going to say, what's the objective function for the best way to visualize something? So let's write that objective function down. I mean, you immediately run into a problem, right? When you're talking about using AI tools, kind of at this very high level of like tying together the experiment theory modeling cycle. Certainly, it has a place at the lower levels of helping through analysis, writing code. But
Could be done with some sort of supervised learning techniques from the literature that we have already available to see what kind of analysis has been done. If we can understand or codify what visualizations are performed and what kind of data that we can go back and automatically suggest a visualization.
I mean, I do think one part, it's a great question. I think we should talk about it. One part of how you've posed it made me think a little bit. You said richer tools or richer analyses. I'm not sure that asking AI to help us with these things will necessarily lead to richer tools, or it'll change things. And I felt like certainly the tools that we've seen today, both software and sort of hardware, right, are already tremendous. I mean, this is this ecosystem, as you all said over and over again, was sort of unimaginable 5 to 10 years ago, that there would be this much support, this many tools, this many very talented people spending a lot of time building the infrastructure for this. How would AI, enrich that? I don't know, but I think it's absolutely worth a discussion. What do you all think? Martin?
models are pretty decent now, but you have many different models, and many different models make many different predictions. Under current data sets that we're collecting somewhat uncontrolled, like we're just showing a lot of random images, many models look the same, but these are in fact different hypotheses, and we can, I think, start to use them to drive the experiment. For instance, you can ask the top two models, which images do you make the most different predictions on? And then you run those experiments. So that data would then tell you and would maximally differentiate between two separate models. So I think bringing the best current hypotheses and competing hypotheses, for me that's in the form of deep network models, bringing those into the experimental design, I think would make this closed loop experimental approach that a lot of you are running a lot more efficient. And that our first paper suggesting empirically that these model-guided experimental data that you collect that way are much better at differentiating between alternative hypotheses.
Yeah, just to throw out an idea of what AI could potentially do. If you have a very large database of data sets with meta information, and people don't even know where to start to begin to look at it, you could perhaps have AI go in there and bring back a summary of what is actually in the data. And then you could ask questions about it and go
Sure.
I want to bounce back on what Mackenzie said. In some ways, I'm very optimistic about the next few years about this. Partly, I'm really inspired by the approach suggested by Blake Richards and Conrad Cording, the novels on neural connectonics. I think I'm less interested in using AI to sort of propose visualization, but more using it as a framework to study what matters. So, Blake Richards and others have proposed, to have the concept of objective function, anatomy, and the optimization algorithm is really important. And on the Pasha shores, we can now monitor up like to 10% of the cells in the mouse cortex. And so we're getting to a point where you can monitor a significant portion of the network where we could see signatures of these three important things, the impact of architecture, the impact of the objective function, and the optimization function. And there must be particular cell types that play a role in implementing this algorithm. And so I think it's up to us to test them. But I think there's an opportunity to collect this massive data set across a large portion of the cortex in the hope of creating and testing this theory, how the cortex might be optimizing certain values. I don't think it's a coincidence that the behavior output is broadcasted. I mean, it must because the objective function is applied everywhere. So I think there are fundamental questions here that we could tackle.
I totally agree with you. I think it'll just be a matter of what task and in what mouse. I think every paper should have said in this task in this mouse for many papers in last year. So there's like the trend of awake behaving animal. It should have been in my task, in my mouse, in my brain area, right?
Yes.
Like I think that's maybe the subtext here. And so like yes, I agree. But optimizing what in what task? Do we need to run the same animal in 10 tasks? Is spontaneous activity enough? And maybe to the point of like the most reused data being the equivalent of quiet wakefulness in a mouse and a human, maybe that's interesting. I just don't know. I would love to see in some sense databases and data also organized that could be large language models that we fine-tune, like Cybert or Great Brain GPT, that we could actually mine data in a smart way around a theory-driven perspective. That's all I'm suggesting. I'm not pessimistic. I'm actually optimistic.
Do we think we can use GPT-like tools to go beyond deduction, essentially? Because when you're talking about theories, you want to, in one sense, you want to do induction of the theoretical landscape. You want to know what theories haven't been explored yet. And so if these transformer-based models are basically remuxing the history of previous knowledge in a sense, then maybe you do still need humans in the loop to some degree or another in order to push that along, right?
Definitely.
ChatGPT is very good to interpolate, but is it good to extrapolate?
Essentially what I was trying to say, right? So if we want to bolster the role of theory, then we need to better understand how to explore the broader theoretical landscape and then use that theoretical exploration to drive modeling, which can then be validated by various types of data that we're talking about.
Yes.
I have another question that's maybe related. which is I'm thinking about kind of the the goal of experiments, and we mentioned the end product is often published in the paper, right? So many presentations were so ultimately more published. And we already know that from the Allen Institute and also from the International Lab Observatory, the data are actually so complex that simply the paper alone are not really gonna cut it. On the other hand, these data sets are very nice, right? They make the data available, but maybe the Allen Institute has made an effort to kind of present the data in this very somewhat user-friendly way where it can be explored. So I'm wondering whether the NIH has thought about kind of the end products and whether publications, there is the data set and there is the publication, and is there some way to kind of have something in between, right? Because for so many of these very complex questions that the brain initiative projects are trying to address, there's only so much information you can put into the paper.
Absolutely, and there are conversations within the NIH about the deficiencies of citation-based metrics based on publications like the H-index and whatnot, and there's been a lot of discussion about that. And there's some more recent history with like software publications describing software releases, and data publications are a trend following on that. And so we are very interested in seeing data publications being a thing, and that becoming a main component of the ecosystem where you can do things like credit assignment, across, nth derivative uses of a particular data set, because that's the thing. We want to incentivize data sharing, right? And we want the scientific community to be happy about it, and so we want people to get credit for it. But that means, if there's a third derivative use, someone used your data set to produce something, and then that data set got used to produce another thing, that some partial credit should redound, to the originators of that data. And so there's kind of a plumbing problem in a sense, making sure all those links remain, that you can roll up that credit, back up to the originators, the full provenance of data as part of the incentive structure of the whole ecosystem. But I think things like data publication and maybe other venues will be important in that. If not.
I was thinking that, going with this question of, we could ask AI to generate the visualizations, right? There's many different ways to visualize the same kind of set of data, and maybe there are some forms for publication that, are not just the paper, but not quite just the data set that could allow for that.
I mean, the Allen has obviously been, field leading in that particular arena. They originally had their own source of funding, right, being completely internally funded and having that as the mission. Very different than how most of us fund our, relatively modest laboratories. But it is something that NIH may want to consider trying to incentivize. That is, how some of the larger brain initiative, the cell censuses are sort of organized. Microns, is, partially with the Allen Institute, et cetera. That has, some similarities to having these extra structures for visualization and data sharing and whatnot. But most of us, in our sort of ad hoc laboratories can't do that. We want to focus on papers as knowledge production, right? We did all this stuff, we spent all the NIH's money, graduate student, postdoc time, et cetera, and at the end, here's this paper, but it represents knowledge. This is actually, something we didn't know before, but then there is all of this data there as well that maybe could be used by us or by somebody else to produce further, knock-on knowledge. How do you think about structures that can be useful for that aren't just, sort of Wild West? Well, we put it out there and there's a ton of tools people can select from and use it. Something more formal, somewhere in between IBL and Allen and, what we're currently doing. I think that's exactly what the point of the symposium is supposed to be doing. So I think it's great. Nima.
So, kind of like building up what Mackenzie said, I think one of the problems is that we're dealing with a system that has too many dimensions. Right? It's just insane. It's astronomical in terms of dimensions. And so when we we sample from them, we always do some sampling. When we are focusing on a given scale, we're always losing perspective of what's happening from the scale. So even if, let's say, if the utility is doing, putting these different electrodes in the brain, then you don't know, for example, what's happening at the level of the individual neurons because you put in these electrodes, but then it's hard to track like what sort of neurons are there and so on and so forth. I think maybe one solution would be to not to think at, okay, let's throw AI at it, but actually go in a way that let's design a set of experiments that the variation in the result, in the outcome, is built into the next set of experiments. Like, what I was saying, for example, we are going to put all these neurons, like, 2 million neurons, and then you still see variations being explained by, later and later component in the analysis that they're using. So this is doing something there. And of course, naively, we're all doing something that is easy for us, which is we are focused on behavior, right? So we all say, all right, let's use behavior because that's the easiest for us to explain this. But, and that's why it's hard to define some metric to say, all right, this is the part of data that we need to keep, or this is the sort of like metadata that we need to describe. I think that's why, because we don't have any theory with it. Reducing it to this sort of like behavior and trying to explain everything from the behavior side, maybe that is the wrong, pattern. Maybe we need to actually change our mindset and go and do something different in that respect.
So I think some principled discussions, based on what do we want to include, how hypothesis-driven do we want to get, and what sorts of test cases do we want to use to potentially produce some of these data sets, and then look, and see, bring some naive users in to try to reanalyze or pull things out. I mean, that to me seems like the way to go to develop some potential processes and sort of guidelines for how to think about doing it more broadly for everybody, potentially. I don't know.
Right, and listening to this, I mean, the thing that I keep coming back to in my mind is that healthy ecosystems are diverse. And so, in a sense, if you start integrating or incorporating this kind of AI-driven output that's based on the history of what's already happened, then maybe you'll be, invidiously leading to some like entrenchment of previous biases, right? And so, in a sense, being, disaggregated and having like atomistic labs, kind of in an ad hoc way, doing their own thing, can sustain ecosystem diversity because there's different approaches being done in each of those labs. And if we think about, okay, we're in this open data era, so what was the major problem with the siloed data era, 20, 30, up to whatever, millennia past? In siloed data paradigm, you've got very deep context. The scientists in that laboratory, have been working with that, apparatus and those scientific questions for a very long time. But then they're disconnected from the rest of the field. So we've increased connection, but we also need to maintain context to some extent. And so that's kind of the main issue that we're dealing with, and maintaining diversity through our tools.
Yes, the back.
So a general thing that I'm kind of taking away is that, we're all trying really hard to find ways to make our data fit into these standardized platforms and paradigms, and they're evolving so quickly, right? We saw today work that is still in progress towards making even better platforms for that. And I guess my question is more from the NIH perspective, do you think an effective or maybe more efficient model might be to have some centralized, group of experts that are funded by NIH to work with labs directly rather than having labs be responsible for keeping up with technology as it evolves in this way to help us get our materials into a standardized format that would be most effective and useful in the field?
I know it probably seems like the NIH is highly in favor of committees of experts.
Not necessarily a committee, but it seems like what I've seen is an effective model in some labs. They have like a professional data scientist that they fund to do this for their lab, right? I have a small lab. I can't afford that, right? So it's either me that does it or a student that does it.
Most labs can't afford that.
Most labs, right?
The vast majority probably can't afford that.
Yeah. So just curious where your head's at.
Well, we shouldn't. I don't think the Brain Initiative perspective is that we're not going to convene a panel of experts to decide what everyone should do with regard to data sharing.
Why not?
Why not? Because experts, well, everyone in this room is an expert on something, and so we all know that we don't know much of anything really. I mean, that's what being an expert is. It's kind of it's not scalable, and it's appeal to authority. It's all the fallacies, and so we want an organic kind of distributed system to emerge, through its own kind of like enabling constraints. Like we want the research community to find, what are the right, protocols and systems and pipelines to put in place.
That does
find what
not
works.
sound like an efficient use of taxpayer dollars, I have to say.
Well, we can encourage people to do all sorts of things. I mean,
Okay, sorry, can I just quickly respond to that? Because somebody gave a talk from the Allen, one of the two talks from the Allen Institute, and it was all about not having the need for an engineer. That if the tools are good, we don't need an engineer to use them. And I think some of the stuff we saw on the slides were so complicated that I thought, I do need an engineer. They're not good.
Yeah, I felt like I needed an engineer to understand the acronyms in some of the slides.
Yeah, I think that would be great, just practically speaking. It's just something on my mind. Not a committee to decide what you need to do, but somebody who's dedicated to help you to meet the objective.
So just quickly, we're in a transitional phase, and eventually, ideally, the technology will disappear into the background, and you won't need an on-site engineer, but maybe that's a good core facility for universities to have.
Oh boy, where are we going to go next? Ali Pasha, we haven't heard from you recently.
Thank you. So I think we are all in this room are pretty much on board and agree that sharing the data is a good thing, and we're agreeing on some of the challenges that are involved and trying to find solutions for that. At the same time, though, I think there is now this mandate by the NIH that we have to upload the data, share the data through these mechanisms, etc. Me as a researcher, I know also a number of our colleagues think that this is not a particularly well thought through decision, and that it is.
It took them 45 minutes, but they have warmed up.
We have a committee of experts.
Yeah, and so because of the financial reasons that were mentioned, so you need it's a cost in time, etc. Because even if we do it, the data will not be probably very useful given the current problems that we are having and so on. So we are just modeling through somehow just to be compliant with least amount of effort that is needed in order just not to be criticized by NIH. Now, I'm interested to hear from you to what extent you think this is a useful thing to do versus what is it that we should be doing, or what are the thoughts? Is the NIH's perspective that the state of affairs as they are right now something that is worth pursuing? Is this data going to be useful?
You can try to ride out the clock?
It's 5:30.
I can see the bar from here. It's not set up yet. You've got a few minutes.
And there's a pit of alligators down there. So I do not represent the NIH, the NIH Office of the Director's DMS policy. I represent the BRAIN Initiative, but the NIH DMS policy says you should abide by program-specific requirements, which means that if you're applying for BRAIN Initiative money or RFA, or we call them NOFOs, Notice of Opportunity, funding opportunity, you obey by our rules, and guess who reviews your data management and sharing plans? It's me. And I am trying to be very, very reasonable, and that's why I often defer to PI discretion as far as where they see the threshold for scientific value as far as downstream uses of their own data sets that they plan to produce. And so in a best effort to be reasonable and agreeable, I can answer further questions if you want.
So based on that, it would be to the PI's discretion if we think that the data is not going to be useful by anyone, just not to upload that. Is that okay?
Well, so we have a lot of RFAs that are based on tool development, like you're just trying to develop a new scope or a new device or whatnot. We don't want you uploading your test calibration data every 6 months. That's not scientifically useful. And so for tool development RFAs, they do not need to submit data according to any of these policies. Though I do encourage that like example validation data sets be shared so that people other labs that want to adopt these tools can make sure that they've installed them correctly and are using them correctly. I think that's very important. And hopefully as a tool developer, that's what you would want to do anyway.
Sandra?
Just a quick thing to just add to this, I think. I know we're at 5:31, but there's a dimension of research software engineers, population that's not often talked about. We talk about grad students, we talk about postdoc, we talk about faculty. We don't talk about a community of research software engineers. And speaking to that point, creating that ecosystem in this country, it exists in other countries in all kinds of ways, would be one way of perhaps satisfying some of the questions that have come up in different conditions. We ask a grad student or a postdoc to do everything.
I certainly do.
They're listening back.
Oh yeah, they're laughing. It's real funny, isn't it?
But maybe that's a way of both kind of encouraging diversity, which NIH talks about, creating pathways for people who are technically really solid and having an opportunity for them to continue in the space of discovery and exploration would be a way. And I know that the DMSP allows for allocation of budget towards this. Maybe at an institutional level that could be aggregated to provide a set of people who could help labs. So that may be a way of a solution to the problem. But I also don't want to over-engineer the solution. We need to solve some of the fundamental questions around why we're sharing data, what we're sharing as well. It cannot just be throw more engineers at the problem.
They're too expensive.
Maybe we should let
Along the same lines.
Shadi, Oliver, and Andrew make their points, Sandra.
Yeah, we
Great.
heard this morning that there are 1,300 or 1,300 brain initiative projects that are funded.
1167.
How much? Let's say
Yes.
1,300. And if we assume that one engineer could support 2,000 labs in helping them with their data, you would need about 54 engineers at a salary of 200K. It's $10.8 million per year. I think it's something worth consideration for NIH to support the repositories that they support with engineers who would work with investigators so that investigators can really focus on the science and get the technical support that they need at a fraction of the cost for the loss, basically, of the intellectual value that they are generating.
There's growing interest in a council of experts, apparently, or engineers. No, that's a, I mean,
It's delegating things to be done by the right people.
I think that,
Let's try to be expeditious.
it's good, quick, but I think just advocating for this. The brain is the most complex piece of matter in the known universe. We're not going to get at this with a little bit more of infrastructure. I'm sorry. At some point, we'll have to sort of, so people have to specialize and develop their expertise in smaller domains. And we can't have the students do everything from end to finish. It makes no sense.
and there should be permanent jobs for people who
Yeah.
specialize.
Who's next, Oliver?
One thing that I just wanted to generally comment is we all hear this ringing in the background of we're expecting perfection from our data when we do sharing, right? But what we need to realize is when we do data sharing, when we develop archive, when we develop standards, we're developing needs for us to talk with each other, and that's just always a decades-long process. And we need to recognize that even the little steps we're making are significant improvements, right? Even if I can just communicate with you how I'm storing a very basic data structure, it's already way beyond what we did 10 years ago, right? And so I think we are making significant progress in what we do. It's just we need to realize we're not going to reach perfection within a year. This is going to take a long time.
Andrew, you want to make the last comment or question? You raise your hand.
Yeah, following up on that, yes, we can't, there's a limited amount of metadata we could have. Any metadata is better than no metadata, and any data online, even if it's a zip file on Dropbox, which I don't recommend, by the way, but that can sit in your filing cabinet in your lab. So small steps.
I'm not sure I 100% agree with you, but I think maybe we'll save that for over a beer, and I'm hoping that'll bleed over into tomorrow.
All right, let's thank Mark and Joe for you.